Imagine a massive, endlessly complex factory.
Okay.
It's running smoothly, churning out, thousands of products a second. But occasionally, a piece of machinery misfires and throws off a stray electrical spark. On a normal Tuesday, it's no big deal. You have safety measures, right?
Right, yeah. Dampers in place, standard protocols.
Exactly. But what happens when the factory overheats, the machinery starts grinding, and suddenly there are furious sparks flying everywhere, literally threatening to burn the whole place to the ground?
I mean, in a situation like that, your standard sprinkler system just isn't going to cut it. You would need a highly coordinated team of internal firefighters. Specialists who can suppress those sparks and salvage the delicate equipment before the structural integrity of the entire building just, well, fails completely.
And today we are taking you on a deep dive into an incredible, largely invisible rescue operation that is happening inside your body right this very second. Our mission today is to uncover how your cells package specialized firefighter proteins.
Specifically, we're talking about heat shock proteins 70 and 90 here.
Right, HSP70 and HSP90. How they package them into these tiny, armored cellular escape pods called exosomes. And they do all of this to shield your vulnerable tissues from the destructive, chaotic forces of oxidative stress.
It is a phenomenal biological supply chain, and to really understand the mechanics of it, we are pulling together insights from some fascinating molecular biology research on complex cellular shipping pathways, biochemical studies on how these proteins act outside the cell, and some very revealing medical pathology regarding the gut and the lungs.
Okay, let's unpack this, because before we can understand the shield, we really have to understand the exact nature of the biological attack. In our factory analogy, those dangerous flying sparks represent oxidative stress. But for the person listening, what actually is oxidative stress on a microscopic level?
It fundamentally comes down to a chemical imbalance inside the cell. So through normal cellular metabolism, just the daily business of keeping you alive, your cells produce byproducts called reactive oxygen species, or ROS.
Which people have probably heard referred to as free radicals, right?
Exactly. Free radicals. These are molecules that are missing an electron. And because they are missing that piece, they are chemically unstable, and they desperately want to steal an electron from, well, any neighboring molecule they can find.
They are essentially molecular scavengers running frantically around the factory floor.
That's a really good way to look at it. Now, usually, your cell's built-in antioxidant defenses neutralize them. They willingly hand over an electron to calm the free radical down, and that maintains a peaceful balance called redox homeostasis.
But I'm guessing that balance can tip.
Oh, absolutely. Under severe stress, maybe you're exposed to harsh environmental toxins, or there's intense internal inflammation or a lack of oxygen. The production of these frantic free radicals completely overwhelms your antioxidant defenses.
And when those free radicals run rampant, they aren't just floating around harmlessly. I mean, they are actively ripping electrons off of things that need them.
Right. They attack the cell's DNA, potentially causing mutations. They oxidize the lipids, which are the fats that make up the cell membrane, effectively punching holes in the walls. Yeah, and they damage structural proteins. It triggers this vicious loop where the damage just creates even more reactive oxygen species.
And we have to recognize where this stress is the most threatening, which is your mucosal barriers. Think about the delicate single-cell linings of your gastrointestinal tract or your lungs. These tissues are the frontier,
They are. They are constantly exposed to the outside world. Every breath of polluted air, every microbial toxin in your gut, every fluctuation in oxygen, it hits them first. Maintaining the physical and biochemical integrity of that mucosal barrier is literally what keeps the outside world from invading your bloodstream.
Which brings us to the heroes of today's deep dive. Heat shock proteins, specifically HSP70 and HSP90. Let's talk about what these are, because normally these are strictly intracellular proteins. They live inside the cell doing their job.
Yes, and they are incredibly old from an evolutionary standpoint. You find these proteins in everything from bacteria to plants to humans. Inside the cell, their primary day job is acting as molecular chaperones.
What does a molecular chaperone actually do, though?
Think about how a protein is made. It starts as a long, floppy string of amino acids. To do its specific job, it has to fold up into a highly complex, very specific three-dimensional shape.
Kind of like microscopic origami.
Exactly like that. Heat shock proteins use energy to bind to these newly formed strings and physically help them fold into the correct 3D shapes. And they also monitor the factory floor. If they find aberrantly folded proteins or proteins damaged by those free radical sparks, they either refold them back into shape or tag them for the cellular garbage disposal.
So they really are the internal firefighters and mechanics. They keep the machinery running.
Yeah.
But the core of our deep dive today is that when things get really bad, they don't just stay inside the burning factory.
What's fascinating here is how the cell prioritizes survival of the wider tissue over itself. Under severe environmental stress, like heat shock or massive oxidative stress, the cell basically hits the panic button. The expression of these heat shock proteins is heavily upregulated. The cell starts manufacturing massive amounts of them. And then, surprisingly, instead of keeping them all inside to fight its own internal fire, it exports them outside the cell to help neighboring tissues survive.
Moving from the inside to the outside is where the biological mystery really begins for me. Because if you look at how cells normally ship things out, HSP70 and HSP90 don't really fit the mold.
Yeah, they don't at all.
They lack what biologists call standard secretory signal peptides. They don't have the normal molecular shipping labels that tell a cell shipping department to put them in a box and send them out.
It's a major hurdle. If they don't have the standard shipping label, how do they get out? Well, they use an entirely unconventional pathway. The cell essentially builds specialized armored vehicles for them. The heat shock proteins get exported inside protective lipid bilayer vesicles called exosomes.
A lipid bilayer just being a tiny microscopic soap bubble made of fats, essentially pinched off from the cell's own membranes.
Precisely. They are literally loading the firefighters into microscopic escape pods. And the mechanics of how the cell decides to load these specific proteins into these specific pods is a marvel of molecular sorting.
How does it do it?
To build these exosome escape pods, the cell relies on a complex piece of machinery called the ESCRT pathway. Within that machinery, a protein named ALIX acts as the crucial loading manager.
Here's where it gets really interesting, because ALIX doesn't just grab things randomly and shove them into the pod. There is an exact structural interaction happening here.
Right.
The ALIX protein has a specific region, sort of grasping arm, called the Bro1 domain. And this arm binds to the tail end of another set of proteins called CHMP4. But it only does this if a very specific passcode is presented.
It is an incredibly precise biochemical handshake. The binding relies on a distinct cone of hydrophobic or water-repelling amino acids. For the ALI-like loading manager to recognize and bind to CHMP4, the CHMP4 protein has to display this specific sequence.
What's the sequence?
A methionine, leucine, or isoleucine, followed by a specific spatial gap of 2 random amino acids, then another leucine, another gap, ending in a tryptophan.
Okay, you just threw a bunch of biochemical letters at me. The research shorthands this as the MLIX at last XW code. It honestly looks like a terrible auto-generated Wi-Fi password.
It does look like a completely random string of letters on paper, but we have to think about this in 3 dimensions. Those letters represent physical amino acids that repel water.
Oh, I see.
Because they repel the watery environment inside the cell, they physically twist and lock together into a very specific shape. It's less like a typed password and more like the physical teeth of a key fitting into a lock.
So that specific physical shape is what tells the ALIX loading manager, okay, this is the right cargo, load it into the exosome.
Exactly. And we know it's a highly intentional, specific lock and key mechanism because there are other very similar proteins in the cell, the CHMP1 through 3 families.
They use the same key?
No, that's the thing. They also have these tail end structures, but they use a completely different spatial pattern of amino acids. Their key has different teeth, which means they bind to entirely different loading managers.
Wow.
This distinct patterning is how the cell exquisitely controls which cargo goes into which vesicle. And during oxidative stress, the cell deliberately uses this ALX pathway to preferentially stuff these exosomes full of HSP70.
But wait, I have to push back on this whole concept.
Okay, go ahead.
I'm still stuck on the fact that this cell is under massive oxidative stress. It's toxic. The free radicals are destroying the lipids and proteins. If the factory is literally burning down, how do we know this isn't just the walls collapsing? Couldn't these proteins just be leaking out of a dying cell through a compromised membrane, and we're mistakenly calling a structural collapse a coordinated rescue mission?
That is the exact skepticism researchers brought to this phenomenon for years. For a long time, extracellular heat shock proteins were dismissed as just passive leakage from dead or dying cells.
Which makes sense on the surface.
Right. But researchers designed some really clever experiments to prove otherwise. They took peripheral blood mononuclear cells, a type of immune cell, and subjected them to intense heat shock.
They intentionally set the factory on fire.
They did, and they watched the exosomes being secreted. They found that the total number of exosomes being secreted didn't change much, but the amount of HSP70 packed inside those exosomes skyrocketed.
Meaning the cell wasn't just falling apart and shedding bubbles randomly. It was actively stuffing more firefighters into the exact same number of escape pods.
Which points to an active loading process. But to absolutely prove it wasn't just membrane leakage or the standard cellular shipping route going haywire, they used chemical blockers.
How did that work?
First, they introduced a drug called Brifeldin A that completely paralyzes the cell's normal protein transport system. If the HSP70 was getting out the normal way, the release should have stopped. It didn't.
The escape pods kept launching.
Then they used another chemical, methyl beta-cyclodextrin, that specifically disrupts lipid rafts, basically scrambling the structure of the cell's outer membrane. If the HSP70 was just leaking through damaged walls, altering the membrane this way should have drastically changed the release.
But let me guess, it didn't.
Nope. Again, the HSP70 release continued completely unhindered.
So it bypasses the normal shipping routes, and it doesn't rely on membrane leakage. It is a deliberate, active, and highly resilient stress response mechanism. Even as the cell is choking on free radicals, it is intentionally loading its internal firefighters into these armored lipid bilayer escape pods and launching them into the extracellular space.
That is the biological reality, and that brings us to the recipient end of this supply chain.
Right, because launching the pods is only half the battle. Now we have to look at what happens when these pods arrive at the neighboring vulnerable mucosal cells, the cells making up the lining of your gut or lungs that are still trying to hold the line. How does this payload actually neutralize the oxidative threat and repair the barrier?
The protective benefits are staggering, and they operate on multiple different fronts simultaneously. First, there is the direct suppression of the oxidative fire itself.
Okay.
When these exosomal escape pods arrive, they are swallowed up by the recipient cells. Once the HSP70 is unleashed inside, it acts as a massive signal booster for the cell's own antioxidant enzymes. It dramatically upregulates the production of heavy-hitting neutralizers like superoxide dismutase and catalase.
So it's not just bringing its own water to the fire, it's handing out better, highly upgraded fire extinguishers to the local defense forces.
Exactly. But it also goes on the offensive to stop the source of the fire. HSP70 has the ability to physically bind to an enzyme complex in the cell called NADPH oxidase, or NOx.
NOx, okay.
Now, under stress, NOx is actually one of the major generators of reactive oxygen species. It's pumping out free radicals. By binding directly to NOx, HSP70 essentially jams the gears, suppressing the generation of new free radicals.
To stick with our analogy, it's actively shutting off the gas line so the fire can't spread any further. That's an incredible mechanism.
It is. And while it's putting out the fire and shutting off the gas, it is also doing some intense psychological work on the cell itself.
Psychological work?
Well, biochemical, but it functions like a psychological intervention. Severe oxidative stress normally triggers a process called apoptosis, which is programmed cell death.
The cell essentially looks around, sees the fire, decides it's a lost cause, and hits its own self-destruct button to prevent the damage from spreading to the rest of the tissue.
Yes, and that self-destruct sequence involves a whole cascade of signals, but HSP70 intervenes. It walks in, pulls the cell's hand off the self-destruct button, and stops the cascade.
How does it do that?
It blocks signaling pathways like J and K, which tell the cell to give up. Most crucially, it inhibits the release of a molecule called cytochrome C from the mitochondria.
Why is cytochrome C so important?
In cellular biology, the release of cytochrome C is the point of no return. Once that's released, the cell is going to die. HSP70 locks that down.
It is literally holding the cell together from the inside, forcing it to stay alive and fight. But keeping a damaged cell alive is one thing. If the mucosal barrier in your gut has been damaged by stress, there are physical holes in the tissue. Bacteria can get through. It has to actually physically repair that tissue barrier, right?
This is where the other major chaperone, extracellular HSP90, steps into the spotlight. When HSP90 arrives on the outside of a recipient cell, it acts as a powerful signaling molecule. It binds to a specific surface receptor on the cell called LRP1.
And what does hitting that LRP1 button do?
It acts as a cellular starting gun for movement. Binding to LRP1 promotes cell migration. This is absolutely critical for tissue morphogenesis and wound healing. It tells the epithelial cells, stop sitting there, stretch out, move into the empty space, and close the physical gap in the tissue.
And we can actually see this physical repair happening in real time in the laboratory. The research highlights studies using a model called DSS-induced colitis. For the listener, this is basically a laboratory model for severe acute intestinal inflammation, kind of similar to what you might see in a flare-up of inflammatory bowel disease. When you expose a healthy gut to this stressor, the mucosal barrier breaks down. The tight junctions fail, and the tissue becomes highly permeable.
Very leaky.
Yeah, and they measure this in the lab by putting a fluorescent dye into the gut and watching how much of it leaks through the lining into the bloodstream.
It's a very clear visual marker of a broken barrier. But when researchers treat that severely damaged, leaky gut with these specific heat shock-loaded exosomes, the recovery is just remarkable. The exosomes trigger the cells to preserve and restore the localization of critical tight junction proteins.
Like which ones?
We are talking specifically about proteins called ZO1 and occludin.
I want to pause on tight junctions for a second because they are so important. If you picture the cells of your gut lining as a brick wall, the tight junctions are the mortar between the bricks. If the mortar crumbles, water and pests get through the wall. ZO1 and occludin are the literal molecular rivets holding adjacent cells tightly sealed together.
That's a perfect visualization. By driving the restoration of ZO1 and occludin back to the edges of the cells, the exosome treatment drastically reduces that mucosal permeability. The cells squeeze tightly together again, stopping the fluorescent dye from leaking through.
So the care packages are literally mixing fresh mortar and boarding up the broken windows.
They are, and it doesn't stop at just the physical repair. The exosomes also trigger the restoration of the biochemical shield. They prompt the cells to start producing antimicrobial peptides again.
Things like defensins.
Exactly, defensins, which act as a chemical moat against gut bacteria. There are even studies looking at duct intestinal cells showing that these extracellular vesicles actively suppress the immune system's panic signals. They dial down the pro-inflammatory cytokines so the tissue can rest and heal.
So what does this all mean?
If we connect this to the bigger picture, it proves that your mucosal barriers rely on two entirely interdependent systems. The physical barrier, made of those ZO1 tight junctions, and the biochemical barrier, which neutralizes toxins and maintains a calm immune environment. Exosomal heat shock proteins are a master key.
Because they regulate both.
Yes, they're uniquely capable of regulating both of these systems simultaneously. They coordinate the physical healing while actively suppressing the biochemical fire.
It really sounds like the ultimate cellular good guy. It's the perfect rescue mechanism. But looking at the breadth of the research we pulled for this deep dive, there's a very dark flip side to this coin.
Yes, there is.
If these exosomal chaperones are so incredibly efficient at healing and keeping cells alive, why are they also strongly linked to debilitating autoimmune diseases and cancer metastasis?
This raises an important question about the fundamental nature of biological signaling. We have to remember that wound healing is meant to be an acute, temporary response.
Right, it's not supposed to last forever.
Exactly. You want the local cells to migrate, you want fibroblasts to activate and lay down some new connective tissue to close a wound, and then you want the signal to turn off. The factory goes back to normal. But what happens if the keep building, keep moving signal gets jammed in the on-end position?
The healing process goes into overdrive and becomes destructive.
Precisely. While extracellular HSP90 is absolutely critical for normal maintenance of the tissue, chronic overexpression completely tips the balance towards severe pathology. Let's look at the lungs. In conditions like pulmonary fibrosis, chronic release of extracellular HSP90 interacts with ongoing cellular stress to promote excessive, non-stop fibroblast activation.
Fibroblasts being the cells that lay down scar tissue and structural scaffolding.
Yes. So instead of a neatly healed microscopic wound in the lung lining, you get runaway scar tissue formation. The tissue becomes thick and rigid, which is exactly what fibrosis is. The rescue signal never turns off, and the over-repair destroys the organ's ability to function.
Wow. And then there's the cancer connection, which is truly sinister when you realize what's happening. The tumors are actually hijacking this exact rescue system to ensure their own survival and spread.
Tumors are chaotic. As a tumor grows rapidly, it quickly outstrips its own blood supply. This creates zones deep inside the tumor of severe hypoxia, a massive lack of oxygen, and subsequently incredibly high oxidative stress.
Which should be fatal.
For a normal, healthy cell, that environment is an absolute death sentence. But cancer cells, particularly the breast cancer cells detailed in our stack of research, have learned to heavily upregulate and secrete HSP90 in response to this hostile environment.
They're essentially setting their own factory on fire but using the firefighter escape pods on themselves to survive the blaze.
They do. The secreted HSP90 allows the tumor cells to survive the oxidative stress that should, by all rights, kill them. But it gets so much worse than just survival.
How so?
The tumor uses that extracellular HSP90 to activate specific enzymes in the surrounding environment called matrix metalloproteinases, specifically MMP2 and MMP9.
And what do these MMP enzymes do?
They are tissue-chewing enzymes. They literally dissolve the extracellular matrix, the structural scaffolding that holds tissues in place.
Oh my God.
Yeah. By using the HSP90 to activate these tissue chewers, the breast cancer cells dissolve the physical barrier surrounding them. This clears a path for them to invade neighboring healthy tissues and escape into the bloodstream to metastasize.
So the very mechanism that evolved to perfectly repair physical barriers and close tight junctions in your gut is hijacked by a tumor to melt healthy barriers down so the cancer can spread. That is an incredibly dark twist, but it really showcases just how potent these extracellular chaperones are. I mean, if someone listening has a relative dealing with fibrosis or cancer, this is exactly the microscopic tug-of-war happening inside them.
It's all about context. The exact same signaling circuit that tells a skin cell to migrate and heal a paper cut tells a breast cancer cell to migrate and invade a lymph node. And this dual nature highlights why targeting extracellular HSP90 has become such a massive frontier in modern medical research.
Because you can't just shut the whole system down.
Right. For decades, oncologists have tried to fight cancer by using drugs to inhibit intracellular HSP90.
But if you shut down the internal firefighters in every cell, the toxicity would be terrible,
Yeah.
right? Because healthy cells need them just to fold normal proteins.
Exactly. The collateral damage is often too high. But the new frontier is targeting the extracellular HSP90, the hijacked form operating on the outside of the tumor. By developing highly specific monoclonal antibodies that only bind to the HSP90 outside the cell, researchers are showing real promise.
That's amazing.
They can strip away the cancer's armor and stop those tissue-chewing MMP enzymes from being activated, all without damaging the normal, essential protein-folding functions going on inside the healthy cells.
It is a phenomenal biological tug-of-war. So to quickly recap the incredible journey we've tracked today. Your cells are constantly fighting off the volatile sparks of oxidative stress. When that stress reaches a boiling point, the cells don't just give up.
No, they fight back.
Right, they pack their internal firefighters, HSP70 and HSP90, into these ALIX-dependent exosomes, using a physical hydrophobic lock and key code to sort them perfectly. They launch these armored escape pods to neighboring mucosal cells, where they suppress free radicals, halt the cell's self-destruct sequence, and physically rebuild the tight junctions to keep you safe.
It is a beautifully orchestrated, highly active defense network. It is the reason your most vulnerable tissues can survive the constant barrage of daily life.
So think about your own gut or your own lungs right now. Next time you take a breath of polluted city air or eat something that gives you a bout of food poisoning, take a second to appreciate the invisible, microscopic rescue operations happening inside you in that exact moment. Millions of cellular escape pods are deploying just to keep your barriers intact.
It absolutely gives you a new appreciation for the hidden resilience of the human body.
It really does. And before we go, here's a provocative final thought for you to mull over on your own. We talked extensively about that specific MLIXXLXXW passcode, the physical key that ALIX uses to selectively load these healing proteins into the exosomes.
Yeah.
If scientists can fully decode and manipulate that exact mechanism in the lab, could we eventually engineer our own artificial exosome?
Oh, wow.
Right? Imagine taking a pill entirely made of programmable synthetic exosomes, loaded with custom payloads of HSP70 and HSP90. We could design them to perfectly target and instantly heal untreatable gut conditions like Crohn's disease, or even perfectly manage oxidative stress to reverse the cellular aging process. I mean, if your own cells can code the payload, maybe we can too.
The therapeutic possibilities of mastering that cellular shipping code are truly staggering to think about.
We hope you enjoyed diving into the microscopic machinery of your own survival today. Keep exploring, keep questioning, and remember that even when the sparks are flying and the alarms are sounding, your body has an entire fleet of firefighters working in the dark to keep the factory running.